Okay, hello everyone. In this video, we are going to discuss about the concept of nucleotide and nucleoside. Okay, there are so many doubts are asked. What is the difference between nucleotide and nucleoside, and what are they? So, in this video, we are going to discuss about nucleotide and nucleoside. Okay, what is this nucleotide? First, we are going to discuss what is this nucleotide. Nucleotide is nothing but the monomeric unit of a polynucleotide chain. Okay, so nucleotide is nothing but nucleotide is the monomeric unit of a polynucleotide chain. In simplest form, if we say what is the nucleotide, then in simplest form, in simplest language, we can say that it is the monomeric unit of a polynucleotide chain or a polymer. What is polynucleotide chain? When more than one nucleotide are attached with each other, then the chain is formed is called polynucleotide chain. And in the polynucleotide chain, suppose this is the polynucleotide chain. Here, this monomeric unit or singular units are called the nucleotide. This is one nucleotide. This is second nucleotide, first nucleotide, second nucleotide, third nucleotide, fourth nucleotide, like this. So these are the nucleotides. And how these are bonded with each other? By chemical bonds. Okay. So when more than one nucleotides are bonded with each other, and the structure or chain is formed, that chain is called polynucleotide chain, or that is the polymer. Okay, so polynucleotide chain is the polymer. And what is the monomer of the polymer? This is the nucleotides. Okay, so what is this polynucleotide chain? When we will say the polynucleotide chain, then this polynucleotide chain refers to nucleic acid, that is DNA, either DNA or RNA. Okay, whatever you are saying polynucleotide chain, then that will be refers to either DNA or maybe RNA, because DNA and RNA are the polynucleotide chains, comes under nucleic acid. Okay, so this is the concept of nucleotide. What is nucleotide? It is a monomeric unit or the single unit of the polynucleotide chain. Then the thing arises, how this nucleotide is consist of. What is the components of the nucleotide? Okay. What is the chemical components of nucleotide? Nucleotide is made up of nucleoside molecule. Nucleotide. Okay. This nucleotide is made up of nucleoside molecule and a phosphate molecule. You may say PO4 minus phosphate molecule. Okay. Phosphate molecule. Okay. So how a nucleotide is formed? Nucleotide is formed when a nucleoside molecule reacts with a phosphate molecule. Then a nucleotide is formed. See, what is the components of, or what is the constituents of nucleotide? Nucleotide is made up of a pentose sugar combined with a nitrogenous base, and this combined with a phosphate molecule. Okay, so this pentose sugar, nitrogenous base and phosphate together called a nucleotide. It is a monomer of a polynucleotide chain. If we does not consider this phosphate molecule, then only pentose sugar and nitrogenous base, these two components, sugar and base, nitrogenous base, are together called nucleoside. Okay, sugar and base together called nucleoside. When nucleoside combined with a phosphate molecule, then that will be formed your nucleotide. Okay, this is the concept. When sugar and nitrogenous base together present, that is called a nucleoside. When nucleoside bonded with a phosphate molecule, then that is called the nucleotide. Okay, then pentose sugar. What does this pentose sugar means? Pentose refers to 5. Pentose sugar means the sugar or the carbohydrate where the number of carbon atom is 5, C5. Okay, pentose sugar, C5. There are two type of pentose sugar. One is, what is the chemical formula of pentose sugar? If C5, CnH2n ON. That means C5H10O5. C5H10O5. It is the chemical formula of ribose sugar. Pentose sugar is generally two type. One is ribose sugar, another is deoxyribose sugar. Ribose sugar chemical formula is C5H10O5 and deoxy. D means remove. Oxy refers to oxygen. One oxygen atom is removed. So the chemical formula of deoxyribose is C5H10O4. If ribose sugar is present in a polynucleotide chain, then that will be called RNA, ribose nucleic acid. If deoxyribose sugar is present in the polynucleotide chain, then that is called the deoxyribonucleic acid, or that is the DNA. Okay. So pentose sugar is generally ribose and deoxyribose, two different types present in the polynucleotide chain, that is the nucleic acid. Then the thing arises nitrogenous base. What is nitrogenous base? There are two different type of nitrogenous base. Generally in nitrogenous base, nitrogen is present in combination to carbon, hydrogen and nitrogen. Okay, apart from the carbon and hydrogen, nitrogen is present. So these nitrogenous bases are generally two types. One is purine, one is called purine, another is called pyrimidine. One is called purine, another is called pyrimidine. So what is this purine and pyrimidine? Generally this purine is double ring compound. Small name, large size. Remember, in this way it will be easy. Small name, large size. That means purines are double ring compound. Double ring compound. This pyrimidines large name, small size. Pyrimidines are single ring compound. Single ring compound. Then this double ring compound, see the structure is like this. This is the single ring compound. Structure of the pyrimidine, single ring compound. But in double ring compound, in purine, it will be like this. Okay, it is the double ring compound. It is the single ring compound. Then what are the examples? What are the examples of purine? There are two different type of purines. There are A and G, adenine and guanine. Adenine and guanine are the two different type of purines, and in pyrimidines there are three, CTU, cytosine, thiamine, uracil. Cytosine, thiamine, uracil are the pyrimidines, and adenine, guanine are the pyrimidines. Then if we compare the melting point, then purines are the high melting point. High melting point and boiling point. Okay, purines are the high melting and boiling point, and pyrimidines are the low melting point and boiling point. So this is the main important thing. What is the structure? Double ring structure. Purines are the double ring structure, and they are two type, adenine and guanine. And pyrimidines are single ring compound or single ring structure. There are three different types: cytosine, thiamine and uracil. In DNA, the combination is A double bond T, adenine double hydrogen bonded with thiamine, A double bond T, and C three hydrogen bond pair with G, guanine. Cytosine present with bonded with guanine. Okay, so this is the general concept of nucleotide and nucleoside. Then if we see according to the base, suppose base A, G, C, T, U, adenine, guanine, cytosine, thiamine, uracil. According to the base, Then what will be their nucleoside and nucleotide? CLE, nucleoside, nucleotide. Okay, adenine. The nucleoside of adenine is adenosine. Adenosine, guanosine, cytidine, thiamidine, then uridine. These are the nucleosides. When they bonded with a phosphate molecule, then they are formed one or maximum three phosphate molecule. Then they will be formed nucleotide. When adenosine converted nucleotide, they are become adenylic acid. They are called adenylic acid, guanylic acid, cytidilic acid, thiamidilic acid, uridilic acid. Okay. When the nucleoside bonded with phosphate molecule, they are formed a nucleotide, adenylic acid. This adenylic acid either may be with one phosphate molecule, AMP, adenosine monophosphate, or ADP, adenosine diphosphate, or maybe ATP, adenosine triphosphate. These guanylic acids like that also may be GMP or GDP or GTP. Guanylic monophosphate, diphosphate, triphosphate. Similarly, these are also CMP, CDP, CTP. These are also TMP, TDP, TTP. Thiamine monophosphate, thiamine diphosphate, triphosphate. These are also uridine monophosphate, uridine diphosphate, uridine triphosphate. So this is the concept of nucleotide and nucleoside. What is nucleotide? It is the monomeric unit of or monomer of a polynucleotide chain. Then how this is formed? A nucleotide molecule is formed when a nucleoside molecule, okay, when a nucleoside molecule bonded with or react with a phosphate molecule, okay, then the nucleotide molecule is formed. That means nucleoside is the component of a nucleotide. Okay, nucleoside is the component of nucleotide. How this nucleoside molecule formed? By the combination of a sugar molecule with a nitrogenous base. That sugar molecule is the pentose sugar, and nitrogenous base is either maybe your purine or maybe your pyrimidine. What is purine and pyrimidine difference? Purine is the double ring compound, pyrimidine are the single ring compound. Examples: adenine and guanine, AG, and their CT, cytosine, thymine and uracil. So this is the generalized concept or quick recap of nucleotide and nucleoside. Okay. So till now we discussed this much. In this video, this much we will discuss. Next discussion on the next video. Thanks for watching, and do like, share, subscribe our channel. Okay. So thanks for watching. Thank you and have a nice day.